Let's say that a beneficial allele has entered into a population, either via mutation or via migration from another population. Then what? Well, typically when an allele enters a population, it will enter the population in relatively small numbers. In fact, there may be only a single copy of this new allele within a population to start with. So what happens to it? Part of the answer is that in all likelihood it goes extinct. And this is because when you have very small numbers, particularly if you have a population where the majority of the individuals in that population are not going to survive until they reproduce, then you will have a high likelihood that any allele that's entering the population, no matter how beneficial, is not going to survive. On the other hand, if the allele does survive in the short term, if it's beneficial, then you will have an expectation that its frequency will increase within the population, particularly if the size of the population is sufficient that genetic drift isn't drowning out the deterministic evolution associated with the higher fitness of this allele. And so you have an expectation that the frequency of the allele will increase, and in fact that frequency will increase until essentially a polymorphism has been formed. A polymorphism here being defined as sufficient numbers of more than one allele that you can actually detect more than one allele at a given locus within a population. If the allele continues to be more fit than the other alleles within the population, its frequency should continue to increase, except to the extent that genetic drift will keep it from increasing, and it will increase until, in fact, the allele predominates within the population. At this point, the allele can then continue to increase, potentially, assuming no frequency-dependent selection against it, until the allele has, in fact, become fixed within the population. At this point, the allele effectively is the only allele that's present at the locus that is associated within the population. So down here you see a frequency of the allele essentially of 0. In the middle you see a polymorphism, predominance, ultimately fixation, where all the other alleles in the population are essentially not present. This is the point at which the allele has in fact been fixed. And beneficial alleles are more likely to become fixed than alleles that are neutral or instead detrimental.